Hello, everybody. So, as I talk about my Sunday class about how we're gonna do this, so I'm just gonna present the last couple of slides, which is 5 lectures, today. So that way, for Wednesday's class, we're gonna be able to do like a little exam review Q&A, and then for Sunday class, which is the day before the exam, we're also gonna have to do a little exam review and Q&A. I just want to get this video out there as fast as possible, just because I know you guys are probably, already stressing about this final. I'm stressing about my finals. So I just want to make sure that we're like, getting there together, I guess, and just making sure that it's as accessible as possible for everybody. Okay. So starting off with lecture 48. So we after lecture 47, we were talking about the initiation of translation. So now this is the elongation of translation, right? Elongation of our peptide bond. So our elongation factors are going to be bringing in our aminoacyl tRNAs that are going to be delivered to the A site. And afterwards, it hydrolyzes the GTP that's involved with our elongation factor. Our actual elongation factors are recycled by another elongation factor. So it's just a whole cycle of elongation factors maintaining the proper mechanism of the ribosome. The interesting thing about the ribosome is that it also acts as a peptidyl-transferase. So it's creating the peptide bonds between these new amino acids. So the amino acid from the P site is then going to be transferred to the amino acid from the P site is transferred to the A site. And that's just the connection, right? So basically what we're looking at right here is basically the amino acid that was in our P site, which in this case is our formylmethionine, is going to have a peptide bond attached to this new amino acid in the A site, and then the tRNA that's in the A site now is going to have two amino acids. So then we're going to have a ribosome translocation, so we need to clear up some of these sites. So using another elongation factor, we're just going to move the whole ribosome to the left. So now the tRNA that was in the A side is going to go to the P side. The tRNA that was in the P side is going to go to the E side. And then our A side is actually going to become vacant, okay, allowing for another tRNA to come in there and bring in a new amino acid. So this is just an image of the translocation. Sorry, it's a little bit blurry. I don't know why it's that. So now how do we terminate, right? So we keep elongating our peptide bonds using the ribosome. So then translation is actually going to stop occurring. And these three stop codons that we talked before are UAA, UAG, and UGA. So we use two release factors, RF1 and RF2, and they recognize different stop codons, but they practically do the same thing. They're going to hydrolyze the peptidyl tRNA bond, terminating translation completely. And then the ribosome recycling factors and our elongation factors G are going to dissociate the whole complex. So in nature of like the termination and of translation and various components of translation, we have to talk a little bit about antibiotics because these antibiotics are going to be targeting different areas of translation. So our puromycin is going to be is going to be mimicking a charged tRNA in the A side, basically inhibiting all translation. Our chloramphenicol is going to inhibit peptidyl transferase. Our erythromycin is going to prevent the movement of mRNA in the ribosome. Our tetracycline interferes with the tRNA anticodon reading, and our streptomycin causes a distortion in the mRNA, so it's read incorrectly. So antibiotics use the properties of translation to stop bacterial infections. We also do have some groups of molecules that actually reduce the expression of some genes in eukaryotes, our microRNAs and our small interfering RNAs. So they have two different functions. So our microRNAs are going to be regulating the translation and the stability of RNA, and our small interfering RNA regulates endonuclease activity. So this is a horrible looking graph, but sadly, you're going to have to know a good portion of it. So for our mRNA synthesis, so we have a pre-mRNA stem loop structure that's broken apart by a Drosha DGCR8 complex. Our pre-mRNA is then exported to the cytoplasm. So our mRNA right here is actually in the nucleus, right? It's transcribed. It comes straight up from the RNA polymerase II. It's transcribed, broken apart by a Drosha complex. Then our pre-mRNA, once it leaves the cytoplasm, is going to be cleaved once more by a dicer. And then the single-stranded mRNA, our siRNA, because they both come from this pre-miRNA, is going to be loaded into an RNA-induced silencing complex, okay? So if we're talking about, if we have an miRNA in this case, right? So basically our pre-miRNA can either become an siRNA or an mRNA once it's cleaved the second time. if we are dealing with an mRNA, when it's associated with this risk complex, we're going to be able to repress translation and increase the breakdown of mRNAs. Okay, and our miRNA actually has imperfect base pairing to the target mRNA untranslated region. That's going to be important in differentiating between miRNAs and siRNAs. And if we have an siRNA risk complex, we're actually going to be cleaving the target mRNA using an endonuclease activity. And our siRNA cleavage requires a perfect base pairing. So if you're looking at the target mRNA recognition, our siRNAs is only going to be dealing with ideal base pairings between the siRNA and the target mRNA, while our mRNA occurs if the base pair is imperfect. So this is two ways that we can recognize if it's an siRNA or an miRNA. So we talked mostly about the translation of prokaryotes. There are some variations for eukaryotes. So the nomenclature is different. So we usually put a lowercase e to represent eukaryotes. So eukaryotes don't use formylmethionine. We use just methionine. Our ribosomes scan the mRNA to look for the start codon in a Kozak sequence, not our Shine-Dalgarno sequence. And the translation, elongation, termination is practically the same thing as prokaryotes. So what are some of our post-translational modifications? So we can have a proteolytic processing. So, we talked about a methionine aminopeptidase in the beginning of this course. So basically breaking apart our proteins. We can phosphorylate it, carboxylate it, methylate it, and acetylate it. The interesting thing is our signal sequence. So this actually identifies proteins that are being made in the cytoplasm. So basically identifies ribosomes that are making a very specific protein, and we can actually add a signal sequence, or a signal sequence is present where we're actually going to identify proteins that that should be synthesized in the lumen of our endoplasmic reticulum. So our signal recognition particle recognizes that signal sequence. So our signal recognition particle is right here. So our signal recognition particle, identifies the signal sequence, and then stops the translation of our ribosomes and moves the ribosomes to the peptide translation complex right here. And in our peptide translation complex, we're actually going to be able to start translating our protein inside the lumen of the ER. And once it's done, our ribosome can then dissociate and move back to the cytoplasm. The interesting thing here as well is that our signal recognition particle, as soon as it brings that ribosome into our peptide translocation complex, is dissociated and can do its job once again. So now lecture 49. So this is all about DNA damage and repair. So this is probably one of my favorite parts of this lecture, just because it's so interesting to see all the different ways our DNA can be so damaged. So we can have a deamination of our DNA bases. So these are some of the ways that our DNA bases can be completely changed. So some chemicals can actually increase this rate of deamination, such as sodium nitrous. We can have oxidation of DNA. So our reactive oxygen species can oxidize our cytosine and adenine, and oxidation can also cause DNA strand breaks. We can have depurination, which is caused by the hydrolysis of the glycosyl bond linking purine to the sugar backbone, and it forms an abasic site. Remember this word because it's going to come back, this abasic site. And we can also have an alkylation of DNA, which is basically a covalent modification of the bases. So we actually create this cross-linking, and we can distort a whole DNA double helix together. Another type of DNA damage is our thymine dimer. So this is caused by UV radiation. Sorry, I can't speak right now. It's caused by UV radiation, and it causes dimerization of our thymines, and it can create these little kinks in our DNA. We can also have DNA strand breaks, which is the worst type of DNA damage, and this is caused by ionizing radiation. It can cause both a single strand and a double strand break. So our cells actually invest a lot of energy just to repair DNA. So DNA even DNA being replicated with any errors can be an ultimate catastrophe. So there are certain phases of DNA repair. First thing, we're going to recognize the problem. Then we're going to remove the problem. We're going to resynthesize that DNA, and then we're going to ligate those loose ends. So you're going to see a lot of the different DNA repair processes are going to follow this type of order. So for a mismatch repair, this is when we're correcting mismatched base pairs, right? So for example, an adenine with a cytosine, as we look at it right here. So the steps are that we have a mut L, mut S complex that recognizes that mismatch. Mut H identifies the parent strand and then nicks the daughter strand. So we're going to find the oldest, which is the parent strand, and then we're going to cut that daughter strand. Okay, so how does mut H know which is the parent strand? So it looks at the levels of methylation. The more methylated a DNA strand is, the older it probably is. Then we're going to use an exonuclease to digest from that nick. So we cut a little portion of that daughter strand, and now we have an exonuclease that's just going into that little opening and eating up a good portion of the DNA. And then our DNA polymerase 3 is just going to go back in there where there's huge gap, fill in the gap, and then DNA ligase is going to seal that nick. So as in this example right here, for example, right? So we see that in this area right here, we have our parent strand, it's up top, right? Because it's the methylation. So we have that nick, right? That we just made. So our exonuclease, in this case, is our 5' to 3' exonuclease. Is just gonna eat it up or eat this up right here, this section right here, right? Since it's a 5' to 3', so it's going from 5' to 3'. So from our 5' to 3', we're going to eat it up, and then our DNA polymerase 3 is going to resynthesize it based on our parent strand that we're using as a template. Then our DNA ligase is going to seal that nick up. For our base excision repair, this repairs mismatch resulting from DNA damage. So the first one could just be a normal replication error. So the steps here is our DNA glycolase is going to cleave the glycosyl bond, creating that a basic site that we talked about before. So eukaryotes have several glycolases, and they all kind of relate to a certain base or a certain combination of bases. Our AP endonuclease is then going to initiate repair by making a single strand break. Then DNA polymerase 1 is going to act as an exonuclease and synthase, and then DNA ligase is going to seal that nick. So our basic excision repair is going to be mainly used for prokaryotes, if I'm not mistaken. So that's why you're going to see we're using DNA polymerase 1. So as we see right here, we have our DNA glycolase. This is our case is our uracil glycolase. We're going to break apart that base. So we just have the deoxyribose that's present here. Once we have the A basic site created, our AP endonuclease is going to break apart, create that little nick. And then our DNA polymerase 1 is just, going to eat that thing up. And then it's going to resynthesize it. And then we're going to ligate it once more. So our nucleotide excision repair, this repairs lesions that distort DNA. So for example, our thymine dimers, for example, or our alkylations. So for prokaryotes and eukaryotes, they have different steps, and you're going to have to know both of them, but they are pretty similar in concept. So for prokaryotes, we have our UVR-ABC exonuclease that makes a nick in the damaged DNA. Our UVRD helicase exercises, so removes a good portion of the DNA and leaves exercise the damaged DNA, leaving that gap. DNA polymerase 1 then fills in that gap, and DNA ligase seals the nick. For our eukaryotes, we have something called our XP exonuclease, which is just a large complex that has XP A through G and our TF2H helicase, if you remember what that was in our transcription, pre-initiation transcription for eukaryotes. So our XP complex scans for kinks, opens the DNA, nicks the damaged strand, and removes it. Then our DNA polymerase epsilon fills the gap, and DNA ligase seals the nick. So basically, UVR-ABC, UVRD, they basically are two enzymes that the XP exonuclease basically does the same function. So XP exonuclease just does a lot of the same things that these UVRs did for prokaryotes. So now we're going to look at our worst types of damage, which is there's a single strand and double strand. So for our single strand annealing repair, this repairs double strand breaks, but this is an error prone repair. So even though we're repairing this double-stranded break, we can still have errors in the DNA because in this case, our cells don't care if there's errors. They just want to repair the double-stranded break because that's the worst damage that they can have, right? So for our steps here, we have our 5' to 3' exonuclease digest the broken ends. Start. So you see the radiation broke it apart. Our 3' to 5' exonuclease just breaks apart just digests some broken ends. Then our single-strand annealing proteins actually bind to the repeat sequences. In this case, it's shown in yellow. Our RAD52 facilitates the base pairing between these two homologous sequences. So then we're actually just bringing them together to match it up. And then our 3' flaps that we're looking at it right here are going to be degraded by our endonuclease right here, our ERCC1 XPF. then DNA ligase seals the nick. So even in this example, you know exactly what error you're seeing because we're basically removing a section of DNA just to repair the double strand break, right? So, radiation broke it apart. Our 5 times 3' exonuclease starts cutting up certain areas of the double-stranded DNA until they were able to identify repeat sequences in which our RAD52 is going to be able to facilitate that base pairing to bring it together, and then we're just going to have endonucleases break apart those little flaps that are still present so that we can ligate it and make it into a double strand break. It's not a perfect repair, but it's the best thing we got. Then the other one that we have is our non-homologous end joining. So this is also for double strand breaks. This occurs at our G0 and G1 of our cell cycle. Oh, sorry, I thought I was going to sneeze. So this is also another error-prone repair, and that's because we're also going to be having those little flaps that are going to be removed. So the steps here are Q70, 80 are going to bind to the loose ends of the DNA. Our PKCS is a kinase that are going to be able to bridge those broken ends, right? Phosphorylating those broken ends. Then our exonuclease will remove the bases in the process to make sticky ends. The sticky ends are basically the same thing that we're looking at right here. They're just going to be able to stick together because they're complementary. So so our exonuclease is just going to be removing bases until we make those sticky ends, but we're also having DNA polymerase mu and gamma, which are also adding bases until we find that perfect sticky combination, right? Then our Artemis endonuclease is just going to remove the flaps once we're able to, put them together, complementary. Then DNA ligase is going to be able to seal that nick up. So now we have to have a little discussion on CRISPR and Cas9. So this is CRISPR and Cas9 was actually used originally for immune system for bacteria, but it could actually be reconverted for genome editing. So what we need to make this complex, we need our CRISPR, which is a clustered, regularly interspaced short palindromic repeats. We need our Cas9 endonucleases, and we need a single guide RNA, which is going to be containing this Cas binding site and a defined genomic target, right? So basically base pairs with the area of the gene that we want. So for the steps, our Cas9 and our single guide RNA are inside plasmids and then are inserted into a cell. The cell starts producing Cas9 and sgRNAs. We're able to form our CRISPR-Cas9 complex, which searches for a target DNA. Our Cas9 then is going to make a double strand or a single strand break. In a double strand, we're actually going to be repairing it by a non-homologous end joining, which is going to be causing random mutations. But for a single strand break, it's repaired by the homology directed repair, which we didn't talk about. But this way, our single strand break, we can actually make synthetic pieces of DNA that can be reintroduced into the new genome. So this is just a picture right here. So this is our CRISPR-Cas9 complex. So I know it's a little bit complicated, but basically you're going to see our sgRNA is going to find a specific area of the DNA that we want. And this is going to allow for our CRISPR-Cas9, especially our Cas9, to make a double strand or a single strand break, which is going to allow for either a random mutation in the case of a double strand break, or a controlled mutation, I guess you could say, or an insertion or a recombination fragment to be added into the new DNA. So basically, sgRNA finds the area of our DNA that we want to, change, and then inside the CRISPR-Cas9, our Cas9 is going to cause a break in the DNA, and then we can insert whatever we want or just let a random mutation occur. So for lectures 50 now, so now this is a little deviation from everything else. So for lectures 50, we talked about signal transduction and cell cycle. So we actually use the mammary gland development as the basis for our understanding of cell cycle for this class. So our mammary gland development occurs in two phases, during puberty and during pregnancy. So the primary hormones that are involved in mammary development is our ovarian hormones, our estrogens and progesterones, growth hormones, and our insulin-like growth factors. So there's two types of signal receptor paths that we're looking at. We talked about a little bit before for exam 3. So our peptide hormone is going to use a receptor in our plasma membrane. And the receptor can be our GPCR, G protein couple receptors, or receptor tyrosine kinases, or it can be a steroid, which just uses nuclear receptors inside the nucleus to act as a transcription factor. So talking about our estrogen, so our estrogen receptor, it's actually inside our DNA. It acts as a transcription factor, basically. we have in a monomeric state in the cytoplasm, it's actually inactivated. But once estrogen binds to it, it induces dimerization. So we get another estrogen receptor to bind with. And this dimeric receptor then moves to the nucleus. In the nucleus, we have a cis-acting element called our estrogen response element. Our estrogen receptor and estrogen complex is then used to activate the genes equipped in our estrogen response element. So once our estrogen receptor binds to the estrogen response element, we're going to bring in, our HATs, our mediators, and our chromatin remodeling complexes, which is going to allow for the activation and transcription of our Jun gene. Okay, remember Jun gene. This is going to be important. So Jun gene is not enough to form memory development. We still need the help of our IGF-1. So IGF-1 is expressed when there's a high levels of growth hormones and estrogens. Uh, and so now that we are able to be producing IGF-1 because it's being stimulated by growth hormone and estrogen, we're actually going to activate it through our RAS-dependent pathway. So we talked about this for insulin. So IGF-1 is going to bind to our receptor tyrosine kinase, causing that autophosphorylation, activates that secondary messenger sequence. And then basically it's going to go practically the same steps that we looked at for our insulin. So our autophosphorylation is going to be promoting this complex. This is going to allow RAS to bind to GTP. RAS GTP interacts with RAF to activate its kinase activity, and then RAF phosphorylates MEK, MEK phosphorylates ERK, then ERK phosphorylate transcription factors. In this case, ERK is going to be phosphorylating our ELK1. So our ELK1, right, we just made our ELK1, is then going to be a transcription activator. It's phosphorylated by ERK, moves to the nucleus. Our ELK1 and serum response factor. Serum response factor is just another way activator for transcription. Our ELK1 and serum response factor are going to create a complex together, and then they're going to bind to an area called our serum response element. So our ELK1 and our SRK are going to be bindings are going to lead to the FOS gene being activated. Okay, so now we have Jun and FOS gene. Now do you have Jun from estrogen and FOS from IGF-1? Our Jun FOS genes are going to be forming into kind of looks like a leucine zipper basically, and they're going to bind to form a transcription factor called activating protein 1. And this is going to be considered an early response gene. So AP1 is really important in starting the whole cell cycle. And it's going to be binding to our TRE, which is then going to induce our MYC and our cyclin D. And these are going to be our delayed response genes. So activation of our MYC target genes. So our MYC target genes, we have our sorry. So what is our MYC genes going to be doing? So they're actually going to be activating pro-growth genes as well. So we actually have a complex of proteins called our MAD-MAX proteins, and they repress pro-growth genes, but our MYC can actually replace the MAD to form a MYC-MAX, which is just an activator for our pro-growth genes. So now that we talked about all the gene regulations for cell cycle, we're going to talk specifically about the cell cycle and all the things that control it. So the cell cycle is basically controlled all the way by cyclin-dependent kinases. And then our CDKs are in turn controlled by cyclins. So we have 3 cyclins that are going to be important to know, or 4, I mean. Our cyclins D and E, which are present in our G1 phase, our cyclin A, which is present in our S phase, and our cyclin B, which is during mitosis. So to start off, the upregulation of our cyclin D gene is caused by the transcription of our AP1 and our MYC, which is just kickstarting the whole cell cycle. The cell cycle ultimately is going to have 2 main restriction points, and the restriction points are basically areas where our cell is going to make sure that the cell cycle is being done correctly. So now looking at more specifically at our CDKs, there are certain inhibitors for our CDK. So we have our INK4, which can actually inhibit our CDK4 and 6, preventing their association with cyclin D. And we also have our CIP, which inhibits the activity of our CDK cyclin complexes. So these inhibitors are basically our restriction points. They act as a wall, especially for G1 through S phase. So only the increased stimulation of estrogen and IGF-1 is going to overcome that wall. Okay. So you're going to see here that our cyclin D is always associated with our CDK4 and 6. So what is happening specifically in G1? We talked about our restriction points, but we didn't really say what happens in G1. So basically, our cyclin D CDK4 and cyclin D CDK6 are increasing. And there's a repressor protein called our RBE2F complex, which inhibits pro-growth genes from being activated. But we need that RBE2F needs to be broken apart for us to move to the next phase. So our cyclin D CDK4, cyclin D CDK6, and our cyclin E CDK2 are actually going to phosphorylate the RB protein of this complex, causing its dissociation and allowing us to move to the S phase. So transition to the S phase. So after we've passed through G1, our cyclin D has no more use, and it can actually act as a repressor towards DNA replication. So cyclin D must be phosphorylated and then broken apart by ubiquination. So our cyclin A must be made as soon as we enter our S phase. So our cyclin A is in a complex with our SYK1, and SYK1 is a repressor of the cyclin A activity. So what our cyclin E CDK2 actually does is phosphorylate our SYK1, causing its dissociation and being broken apart to allow cyclin A CDK2 to do its job, which in this case is DNA replication. So now moving to, actually, let me just take a drink of water. Oh, I forgot, I don't have water. I had like 3 drops. So now that we talked about our cell cycle, now we're going to talk about our cancer biology. So what makes a tumor cancerous? It has to be both invasive, so it can break through other tissues, and metastatic, which is being able to colonize new sites of our bodies. So these are some of the new terms we're going to be talking about. We have a proto-oncogene, which is a normal gene that encodes for cell growth, but has the capability of becoming a cancerous gene. We have an oncogene, which is just a mutant proto-oncogene that favors excess growth. And we have an activation, which is just a conversion of a proto-oncogene to an oncogene. So there's 2 types of class of tumor. We have a sporadic tumor, which is a tumor that has no inherited genetic defect, and a genetic predisposition tumor, which is an inherited defective gene. So how do we activate, convert basically from a proto-oncogene to an oncogene? These are the 3 ways that are, or these are the 3 ways that are presenting in class. We're only going to be talking about 2, but basically we can have a mutation to coding sequence, we can have an amplification of a gene, so basically an overexpression of a normal protein, or we can have a chromosomal rearrangement that causes the overexpression of a normal protein. So the first thing we're going to talk about is a gain of function mutation, which is referring to this mutation area right here. So there's 3 forms of gain of function mutation. So this is the first one is when we're actually increasing the signaling for cell growth and proliferation. So remember we talked about our RAS dependent pathway. So we're going to go a little bit more detail into it. So this actually occurs at our HER2 protein, which results in a dimerization. So basically a mutation in this receptor right here results in a ligand independent activation. So we don't need a ligand floating around the extracellular fluid to activate this receptor. And it's always going to be in this activated state, meaning the pathway is always activated when it shouldn't be activated. So kinases can also be a target in therapy against cancer. So the most important one to note is our Herceptin. So this is actually a synthetic antibody which recruits the host immune cells to kill tumor cells that overexpress this HER2 receptor protein. So our second gain of function mutation, this is when our RAS can be activated in the absence of a growth factor signaling. So a RAS mutation, remember our RAS is associated with GTP. A RAS gene mutation can result in the RAS protein not being able to hydrolyze that GTP, leaving in this perpetual activated state. So this is a picture right here. So we see that once our activated RAS is one associated with GTP, but if it's unable to hydrolyze back into a GDP form, which is this inactivated form, it's going to continue activating our RAF, which is going to continue activating that pro-growth pathway. And then finally, our third gain of function mutation is our RAF can become hyperactivated in the absence of growth factors. So this is just a mutation of our actual RAF. So let me just go back here because I just want to make sure everybody. So this is what we're talking about again, right? So we had a mutation in our actual receptor. This is not the same receptor, but it has a similar purpose. We saw one mutation in the receptor, one mutation in our RAS, and then one mutation in our RAF. And all of these, is if one is broken apart, the whole pathway can become completely activated and uncontrollable. So the other way that we can look at these mutations are chromosomal rearrangement. So this is another type of activation pathway. Some of the common abnormalities we can see is our aneuploidy, our deletions, duplications, inversions, insertions, and translocations. So the interesting thing that we like looking at for these chromosome rearrangements is our Philadelphia chromosome. So we have two different chromosomes right here. We have a fused ABL gene from our chromosome 9 and our BCR gene, our chromosome 2. So we can actually have a translocation where our ABL gene moves to chromosome 22 to form this BCR ABL functional protein. The issue is that ABL is actually a kinase, and this translation causes a hyperactive tyrosine kinase receptor activity. So we're activating signal pathways without stopping because this combination of chromosomal rearrangement actually forms a functional protein that causes this hyperactive tyrosine kinase activity. Looking into more, we also have our enhancer hijacking in our medulloblastoma. So medulloblastoma accounts for a large percentage of our pediatric brain tumors. So this actually is an overexpression of our GFI1B transcription factor, or I'm sorry, this is done through chromosome rearrangement, and one of the symptoms, one of the signs, is an overexpression of this transcription factor, which is pretty common. So you actually see it can actually have two different ways that a chromosome rearrangement can occur. We can have an inversion of our chromosome to allow our DX31 and GFI1B to be really close to each other, causing a problem, causing a mutation, causing a chromosome rearrangement that causes cancer, or we can have a huge deletion of our chromosome, allowing our DDX31 and our GFI1B to be close again, causing again this chromosome rearrangement to become cancerous. So for retinoblastoma, there's two forms. We have our sporadic with just random mutation. So you actually inherit two good copies of our RB gene. It just was a random event that you got cancer in your eye. Or we kind of have a predisposition where our patient already has one defective copy of our B gene. So basically looking at a retinoblastoma and our pedigree, we actually hypothesize a two-hit hypothesis. So basically the activation of one single copy of a tumor suppressor gene will not cause the cancer, but a loss of a second one will do it. So we need two defective tumor suppressor genes to cause cancer. So defects in chromosome segregation, mitotic recombination or mutation can lead to that second hit, even though you might have a predisposition already. So the most important tumor suppressor gene is our P53. This is inarguably the most important one, the most studied. So this is activated in response to cell stress and DNA damage. So it controls DNA repair, cell cycle arrest, and apoptosis. Apoptosis is programmed cell death. So when our cells actually just basically commit suicide. So mutations in our P53, for the first hit, our P53 is still going to work because we have a second copy of it. But for a second hit, it will cause a decrease in the functionality of P53. And some mutations have a dominant negative phenotype that causes a mutation or a zinc finger motif as well. So as we look at in this image right here, so our P53 is just a classical zinc finger protein that binds to DNA. And it's encoded by a TP53 gene. So if we have one hit that shows right here, we're still transcribing a bunch of our important tumor suppressor genes. However, if we have two hits, we're going to cause a mutation in an epigenetic silencing, causing a lack of transcription of other tumor suppressor genes and other genes that are important, for example, in apoptosis, G1, G2 arrest, and DNA repair. Sorry. And then, as I said before, we can also have a dominant negative phenotype. So some missense mutations, so adding another amino acid into our protein can have this dominant negative phenotype, and this causes a mutation that affects our zinc finger motifs that doesn't allow our other, proteins and other important molecules that we need to be made from being able to be made, even though we have one functional gene already present. So, our human papillomavirus, or HPV, is actually one of the cancers that is brought about by viruses. So that actually makes cancer pretty communicable. So our HPV has two important proteins, our E7 or E6, and these are the proteins that actually sequester our P53 and RB, and these are two very important tumor suppressor genes. So as we look at right here, our HPV 76 can actually bind and stop the work of our RB and P53 by basically inactivating them. So if we look at an image right here, so for example, if this was working properly, our cell proliferation would be blocked by our P53, which activates the safety brakes in our cell proliferation, and our RB, which is also preventing cell proliferation from occurring. But our viral proteins, in this case, our E7 or E6, can actually bind to our RB and P53, allowing cell proliferation to occur without any stops, basically. Now talk about a little bit about breast cancers. So these are defects in our BRCA1s and BRCA2s, and these genetic defects are actually inherited, and 80% of daughters with these defective genes will actually get cancer. So BRAC2 works is a mutation that works in our homologous recombination repair. BRCA1 is still undefined, but we also think it's pretty closely attributed to DNA repair mechanisms. So if we have a defect in DNA repairs, you know that we're going to eventually have a lot of mutations because our DNA isn't being repaired and properly maintained. So for tumor progression, as we're going to see, as one cell undergoes mutation, this leads to all their daughter cells having even more mutation and more mutation and more mutations, causing this huge increase in cell mass that has no real functionality and can become cancerous eventually. So finally, actually for my last slide here, so for our tumor angiogenesis, so this is how our tumors are actually able to build their own vasculature system for survival. So tumors activate pathways for blood vessels to be growth, and they usually are vascular endothelial growth factor, or VEGF, and our fibroblast growth factors to be activated to cause blood vessels to surround the tumor and allow the tumor to survive. And how does the tumor become invasive? So how can it reach other tissues or break apart other tissues? So basically, our epithelial cells have something called our anoicas, which our anoicas basically is a process, sorry. So our epithelial cells have these attachments to the basement membranes. And when we go through anoicas is when our epithelial cells actually lose the attachment to the basement membranes when they are killed. And tumor cells must be able to uh, must be able to go through synoikis to acquire this invasive behavior. So it must be able to break apart the epithelial cells that are attached to the basement membrane to acquire that invasive behavior. Right. So this was actually the last lecture we're gonna go before for final. Um, I'll be putting more stuff in GroupMe just for how this week's gonna look like. But be sure that for the next sessions, we're just going to be going over reviews and some questions and just going over last minute things if you have any questions. Okay.